Hi, hello, namaste, very good evening to all. Myself Murali PG. Today, let us have one YouTube class under Unacademy where we are going to have the discussion of most important biology questions for second PU board exam. Regarding with this, I am taking today's class. So let us discuss the second part of questions, model questions. Okay. So with respect to Unacademy for Karnataka CET also, we introduced some of the courses. There is a plus course where it is with the following important features: premium content at affordable price, structured live courses, quizzes at regular intervals, batches for complete preparation, test series for DPPs, full length mock test, and analysis, single subscription for unlimited access. So everything is under single plus subscription. And with respect to our Unacademy class or the concept, 20 plus top educators are taking the class, 10 plus batches are there, 250 plus courses, 2000 plus lessons are added, and many more added every day. So use my code KCET Murali and get 10% off during the time of plus subscription. You have to do one thing: download Unacademy Learners app, install it, and you can take it from Google Play Store and install in your mobile or in a laptop. And under search go to Karnataka CET, go to state CET under which Karnataka CET, and you can have the selection of plus course categories. Okay, so during the selection you can use my code KCET Murali and get 10% off. These are the price values for this month, and you during that time subscription, use my code and get 10% off regarding with our plus classes. So Unacademy is going to give you all the required necessary things for you to prepare for KCET and also the board exam. Already the dates were announced under Karnataka CET and Karnataka PU board. So here the dates are given for the board exam timetable and tentative timetables, and for CET also. Okay, there is another practice test series for Karnataka CET Aspirants where daily test 20 questions in 20 minutes. Take live classes, get leaderboard ranking, evaluate your performance, attempt later multiple times, and practice regularly and gain confidence for exam. So it is available for free, and you can make the use of it, and you can get the confidence to face the oncoming exam. So by this brief introduction, so this is how to attempt a test series. Okay, so here important question set second for reduced syllabus 2021. As we all know that due to Corona COVID-19, here the board exam, PU board, Karnataka PU board reduced the syllabus for board exam. So according to that, we prepared here. I am giving some of the important questions. So in the previous class on 23rd, I already taken the first set questions, and here the second set of questions today let us discuss. Okay, under board exam category, and the next class will be on 27th. There also we are going to have the important question set 3. The third question sets are going to be discussed. Okay, my dear friends, very good evening to all. Welcome to the class. Let us begin with our question. So part A, part B, part C, part D. Yes, we all know that 1 mark question, 2 mark question, 3 mark question, and 5 mark questions with choice. So remember, for 1 mark question there is no choice at all, isn't it? So here let us have the questions. For example, what is this first question? Yeah, which is the first question? Define ovulation. So ovulation means what? The Graafian follicle ruptures to release the secondary oocyte, that is the ovum, from the ovary, and it is lodged at the amphilisthmus portion of the fallopian tube. This process is called ovulation, or simply we can say it is a release of ova from the mature Graafian follicle. So as a result of that, secondary oocyte is going to be released from the Graafian follicle. That is what ovulation phenomenon. So what is meant by ovulation. Okay. And second question if you take, what is polyembryony? What is meant by polyembryony? Presence of poly means many, embryo means you know what is embryo, present inside the seed, isn't it? So what is polyembryony? Many means here, many word means the presence of more than one embryo. Usually in one seed one embryo is there, monoembryo, isn't it? Actually it is a normal condition in the case of our angiosperm. Coming to the point of some of the example for angiosperm, here with respect to mango, the polyembryony condition is there. That means more than one embryo is in a seed. So one seed contains more than one embryo. That is called polyembryony phenomenon. Then what is emasculation? The third question. So it is a controlled pollination mechanism. So removal of stamens in a bisexual flower before its dehiscence to prevent self-pollination, that is called emasculation. So removal of unwanted stamens to overcome the unwanted pollination. So removal of stamens to overcome the unwanted pollination means here controlled pollination and fertilization. Emasculation is one of the technical aspects. Yes, Manoj, welcome to the class. So we are in a discussion of 1 mark questions. Okay. Next, fourth one. Why the amniocentesis is banned? Amniocentesis. Amniocentesis refers to study of chromosomal pattern observed in the amniotic fluid of the fetus in the developing stage. So why this amniocentesis is banned? Why it is banned? Because here it is a statutory ban on amniocentesis for sex determination to legally check increasing the chance of female feticide, massive child immunization, etc. So mainly to minimize the killing of female fetus, the amniocentesis is banned. Then define polygenic inheritance. What is polygenic inheritance? Coming under genetics chapter, an expression of a trait is controlled by 2 or more number of gene. Expression of a trait means a character is controlled by more number of gene is called polygenic inheritance. It is not pleiotrophism. Pleiotrophism is different from polygenic inheritance. So here it is the, yes Manohar, welcome to the class. Must say it is the expression of trait is controlled by 2 or more number of gene is called polygenic inheritance. Then what is linkage? Linkage and recombination in the case of T.H. Morgan experiment under genetics. So it is the tendency of 2 or more genes to remain together in the original combination on the same chromosome from generation to generation. That is the linkage. Or in another way we can explain what is linkage is the physical association of genes on a chromosome. It's a simple explanation. Isn't it? Physical association of gene in genes on a chromosome. Yes Manohar, what is that? Yeah, not 10 mark. 10 mark means 1 mark 10 questions. Okay, 1 mark 10 questions, important questions. Okay, here today one second part. The last class I taken the first part of the set. Second part is today, and third third or set third will be there on 27th. Okay, yeah. So what is linkage? Linkage is the physical association of genes on a chromosome, that is called linkage. Then which chromosome of humans has the most number of genes? So coming under Human Genome Project, the outcome of Human Genome Project, or what is the main output of the Human Genome Project? One of the thing is the knowing of number of most genes present in a chromosome. So which is that one? Chromosome number 1. Chromosome number 1 is having most number of genes. Then name the type of antibody produced during allergy. During allergy, as we all know that it is IgE. Immunoglobulin E is produced at the highest level. Okay, yeah. Then what are genetically modified organisms? GMOs ಅಂತ ಹೇಳ್ತೀವಿ. What is GMO? Organisms. It may be bacteria, fungi, plant or animal, whatever, whose genes have been altered by manipulation through genetic engineering. That is called genetically modified organism. Modification in their genetic level. Modification of the organism in their genetic level. Organism may be plant or animal, fungal, bacteria, etc. Okay. Then mammals from colder climate have shorter ears and limbs. Why? Justify your answer. Why it is there? Why that kind of physiological, morphological feature is there, isn't it? So mammals from colder climatic generally have shorter ears and limbs to minimize the heat loss. So to minimize the loss of heat, the animals which are found in the colder climate have shorter ears and limbs. Yes, Arjun, welcome to the class. Hi, namaste. Okay, these are the 1 mark questions. Part A 1 mark question. So I prepared here according to part A, part B, part C, part D. Part A 1 mark, part B 2 mark, 3 mark part C, and D is the part D is with the 5 mark question. Now let us continue with our next set of question. Part B. As we all know that here we can observe nearly about how many questions? 2, 4, 6, 8. Am I right? Yeah, 8 questions will be there. Out of 8 you have to answer for 5, isn't it? So it is according to the rules and regulation of our PU board and CIT. Now coming to the question number 11, let us have the questions now. Differentiate between staminate flower and pistillate flower. Staminate flower and pistillate flower. Staminate flowers are male flowers, are female flowers. Staminate flowers are male flowers and pistillate flowers are female flowers. Then mention the accessory duct. Yes, am I right? Yeah. Mention the accessory duct of male reproductive system. Primary sexual organ of male is the testis, and it is associated with the accessory duct. Which are the associated duct? Accessory ducts that includes rete testis, vasa efferentia, epididymis, and vas deferentia. So that is coming under human reproductive biology, isn't this question? Then 13th question. List any 2 principles to be followed to prevent the sexually transmitted infection or sexually transmitted diseases. What are the 2 important principles we have to follow to prevent the STDs? Methodology. Avoid sex with unknown partner, multiple partners. Always use condoms during intercourse. In case of doubt, one must consult a doctor, qualified doctor, without delay. So these are some of the preventive measures with respect to avoid the transmission of AIDS, HIV, kind of sexually transmitted diseases. And next 14th question. So this is from the reproductive health chapter, isn't it? 13th question. Got it? 13th question, it is from reproductive biology, reproductive health. Okay. Next 14th question. Write the karyotype of Down syndrome and Klinefelter syndrome. What is this karyotype? Means genetic representation, chromosomal representation of Down syndrome. So as we all know that Down syndrome is what? Autosomal disease, isn't it? It is because of autosome disorder. So here we can observe addition of 1 autosome. So 45 will be there instead of 44. 45A plus XX or 45A plus XY. That is in the case of male. This is in case of female. So autosomal chromosomal disorders will going to occur irrespective of gender in case of both male and female. That is a Down syndrome. Coming to the Klinefelter syndrome, it is confined only to the male, where 1 additional X chromosome is there. How to represent? 44A plus then XY. 44AA plus XXY, one additional X chromosome. So this is the Klinefelter syndrome. Like the Turner syndrome, we can mention how 44AA plus X0. It is observed in the case of female. One X chromosome is less. Here one X chromosome is addition in the case of Klinefelter. Klinefelter syndrome is confined to the male. Okay. Turner syndrome to the female. Okay. Next question. How anaerobic bacteria are beneficial in secondary sewage treatment? So microbes in human welfare chapter. Isn't it? Microbes in human welfare chapter. Anaerobic bacteria are beneficial in secondary sewage treatment. Primary sewage treatment. Primary treatment is there, followed by secondary treatment. So here anaerobic sludge digester, as we all know that concept, anaerobic sludge digester. There we can observe the kinds of bacteria which grow anaerobically, digest the bacteria and fungi in the sludge. During the digestion, bacteria produce biogas. That biogas can be utilized for source of energy as it is inflammable. So it is what our importance. So use of microorganism in the field of sewage treatment. This concept is coming under microbes in human welfare. Okay. Yes. Next question. Which are the four basic processes that can fluctuate the density of population in a given habitat? It is very simple. Natality, mortality, immigration and emigration. Natality means birth rate, mortality means death rate, then immigration and emigration. Immigration means flow of number of individuals from other population to our interested population. It is immigrants. And emigration means the organism will move from our interested population area to the other one. That is called emigration. So death rate and emigration will going to decline with respect to population percentage, whereas natality, that is a birth rate and immigration will going to boost the percentage of population density. Yes, the next question. What is sacred groups? Give an example. Biodiversity and conservation chapter. See, ecosystem ecology chapter. In that ecology chapter, two chapters were deleted, isn't it? Organism and population and biodiversity and conservation is retained. But the other two chapters, that is our ecosystem and environmental issues chapter, these two are not included. Yes, Shishika. Yes, this is some of the important question what I worked out from last several years question paper. So I am giving this one in the form of set 1, set 2, set 3. Set 1 I already discussed in the previous class, that is on 23rd. This is second one, set 2, and the third one that is on 27th. 27th I am going to give the third set of questions and also diagram based questions. Okay, that also going to be discussed. Okay, answer you know what is this questions and answer, but which are the most important questions, commonly asking questions. Yeah. Okay, Shishika, got it? Yeah. What is sacred groves? Sacred groves are one of the type of in situ conservation method, isn't it? A group of trees is referred to as grove, and groves are considered to be God's first temple. So no one is allowed to cut a tree or kill the animal in that grove. So you can observe Deodar grove in Shimla. So in our local Manoj, important diagram, so that is on next class. Okay, that is on 27th. I am going to include that one also. Okay, the 27th another set of questions along with the diagrams, important diagrams. Okay, yeah, today only questions. Yeah, here also, here and there the questions will carry the diagrams, but only for diagram I am going to take on 27th another along with another set of questions. Okay, yeah. Sacred groves, you know what is that. Then alien species invasion leads to the extinction of indigenous species. Justify the statement with the two examples. So here Nile perch introduction result in the cichlid fish extinction. Then Parthenium and Lantana and water hyacinth pose the threat to indigenous species of that aquatic ecosystem. If you take water hyacinth, Yeah, diagrams that I will going to take in the next class. Okay, yeah. So alien species invasion leads to the extinction of indigenous species. Justify the answer. So here also there is the organism and population in that it is coming. Next one, part C, that is 3 mark questions. Let us have the 3 mark question discussion. So here how many questions are there? 1, 2, 3, 4, then 5, 6, 7, 8. Out of 8 you have to answer for 5, isn't it? Yeah. Draw a neat label diagram of monocot embryo. See, here diagram came. Shashika, Manoj, got it? Yeah. So once again we will going to discuss diagram based questions in the last class, in the next class, that is on 27. Okay. So here label diagram is we have to give the clear picture of that diagram along with the correct labeling. That is important. Please use half page of the answer sheet. In the entire page you should use half page to draw the diagram, a clear diagram with the labeled part. So labeling should be correct. Okay. So for 3 mark questions, minimum 6 labeling is needed. It is better to label 6 parts if it is there. Okay. For monocot embryo, of course, nearly 6 to 7 parts are there. Okay. Then write the characteristics feature of insect pollinated flowers. So characters of flowers pollinated by insect. So direct characters, 4 characters or 3 characters will be there. So it is pollination by entomophily. So coming under type of pollination, mechanism under sexual reproduction flowering plant. Okay, make a note of it. I will tell where actually it is coming in which chapter. Okay. Then what is parturition? Briefly explain the process of parturition. So here the process of labor, the process of childbirth, that is the parturition, isn't it? Vigorous contraction of uterus at the end of pregnancy causes expulsion or delivery of baby, that is what? Parturition. So you have to explain briefly with respect to parturition, which is the hormone going to be produced, oxytocin, then fetal ejection reflex phenomena. So all those things we have to write briefly. So that is another important question coming under human reproductive biology. Then how can conception be prevented without the use of contraceptives? See, this is a natural method of prevention of unwanted pregnancy. How can conceptions be prevented without the use of contraceptive? Natural method. See, directly we can ask, write the natural method of prevention of contraceptive, prevention of pregnancy, or preventive measures of conception. Instead of that, here we twisted the question, like how can conception be prevented without the use of contraceptive? That is what we have to explain. Natural method of preventive measures for the conception. This is how the question will be there. So please read the question properly and try to understand the meaning. Then you can answer. Of course, you know the answer, but without understanding the question, it is very difficult to answer, isn't it? So a lot of students are getting the facing the same difficulties. I know. Next one. So here natural method of periodic abstinence, lactation, amenorrhea, isn't it? Periodic abstinence, lactation, amenorrhea, coitus interruptus, and 3 methods are there to overcome the unwanted pregnancies under natural method. Then see one long sentence. DNA polymorphism is a very useful identification tool in forensic applications. Mention the steps involved in identification of polymorphism using DNA fingerprinting technique. Means what? Here nothing but what is that satellite? What is the guidelines for that DNA fingerprinting? Means methodology for the DNA fingerprinting, and sequencing, how it is going to be done depending on the base composition, ATGC sequence, then satellite DNA usage. So that is what we have to write. The major methodologies followed in the DNA fingerprinting technology. Okay, so it is an application part of molecular biology, and it is needed in today's lifestyle, isn't it? And next question number 24. Okay, with reference to malaria, answer the following. See, splitting of questions. Here this is for 3 marks, isn't it? 1.5, 1.5. Name the pathogen and vector. which is a pathogen for malaria? As we all know that Plasmodium. In that three types: Plasmodium vivax, Plasmodium malariae, Plasmodium ovale, and Plasmodium falciparum. So four types are there, isn't it? Plasmodium falciparum. Plasmodium is a general pathogen. Then vector is a, you should write female Anopheles mosquito. Don't write mosquito. Don't write Anopheles mosquito. Write female Anopheles mosquito. Only the female mosquitoes will going to suck the blood, not the male mosquitoes. Okay, because to lay the eggs and to complete the life cycle with respect to female mosquito, it requires blood. Okay, so give the authentic answer, give the correct answer, shoot the correct answer. Don't divert the answers. Okay, name the pathogen. Plasmodium, scientific word. Vector, Anopheles female mosquito. Okay, next, symptoms as we know: higher fever, chilling, fever occurs only every alternate on every alternate day, then vomiting, other things, so that we know. Then list out any three types of innate barriers of defense with example for each. Innate barrier under human health and disease chapter: physical barrier, physiological barrier, cellular barrier, cytokine barrier. As we all know that, isn't it? That is the answer for the question number 25. Then 26, once again, another, draw a neat label diagram of simple sterile tank bioreactor. Bioreactor under biotechnology process. So use of bioreactors, use of bioreactors. Simple sterile tank bioreactor diagram you should study, and you have to draw with the labeled part. Okay, then yes, of course, I am coming to the next important part, 5 mark questions. In that, section 1 and section 2 is there. Second section 1 and second. Okay, so in that, first see, in the 3 mark question, minimum 2 diagrams or 1 diagrams will be there. 1 diagram will or 2 diagrams will be there. In the 5 mark question, once again 2 diagrams will be there. So please use of it, and if your drawing is very good with the label part, of course you will get a full mark. So minimum 15 mark diagrams are there. Okay, yeah. Then coming to the point here, draw a neat label diagram of mature embryo sac of angiosperm. Mature embryo sac of angiosperm, what is that? Our 7-celled 8-nucleate structure embryo sac, isn't it? Antipodal cells, egg cell, synergies, and central cell with the polar nuclei. So all those things should be there in a proper level. So we will discuss that our diagram based questions, of course, in the next class separately. Okay, then explain Mendel's experiment of inheritance of one gene with reference to height in a plant or pre-plant. So see here, explain Mendel's experiment of inheritance of one gene with the reference to height in the pre-plant. We can ask this question in another way. Mendel's first law. Explain Mendel's first law, or define Mendel's first law with the example, or explain Mendel's experiment of inheritance of one gene, one character hypothesis. That is also same. That is what tall into dwarf. Tall into dwarf, what we are getting, 1:2:1 or 3:1 ratio. So please use that one in a proper way. So try to understand, and we can make that question in another way. One gene, one character hypothesis, Mendel's first law with the example. Okay, otherwise like this. Then draw a neat label diagram of sectional view of mammary gland. Yeah, mammary gland sectional view. That is in the case of human reproduction chapter, mammary gland sectional view. That is for 5 mark. Remember, yeah. Then list the salient features of human genome. What is human genome? Human genome project is different. Human genome. So salient features of human genome. Okay. So 30th question, am I right? Yeah. Yes, list the salient features of human genome. So in that you have to mention what is human genome, how many nucleotide bases are there, then 3000 bases every gene, which is the largest gene, that's the dystrophin. Total number of genes estimated is 30,000. 99.9% nucleotide bases are same in all people. Then 50% of gene discovered is unknown. Less than 2% gene is going to code for protein. Repeated sequence make up a large portion of human genome. Chromosome 1 has most genes. See here, in one of the 1 mark question I said, I mentioned this one. What is that? Yeah, which chromosome of human has most number of genes? That is chromosome 1 here, and with the gene 2968 genes. Okay, and least is in Y chromosome, isn't it? In Y chromosome, where we can observe how many genes? 231. In Y chromosome 231. In chromosome number 1, 2968 genes are there. So these are the salient feature of human genome. So we can ask this question as write the salient feature of human genome project. That is also same answer. Okay. Next question number, right? Yeah. Next question number. What is the question? Yeah, lack of operon. Of course, very interesting part, isn't it? Lack of operon. Explain the regulation of lack of operon in the absence of and presence of lactose in the medium. We can observe. We can ask another question directly. Lack of operon. Explain lack of operon. Another question is negative feedback mechanism. So negative feedback mechanism that is also going to be explained by using the lack of operon model. Okay, so that is one of the important question. So switch off model and switch on model. So what is the yes sir, Manoj, lack of operon is very important, because in old syllabus lack of operon, histidine operon, tryptophan operon, valine operon, so other types of operons. In under 2 mark question we can ask name any 2 operons. So lack operon, histidine operon, tryptophan operon, valine operon. So these are the things. Okay, in other examples for operon. Okay, then 32nd question. So here we have to draw the diagram. Okay, lack operon diagram, schematic representation diagram, that is important. When the lactose is present, when lactose is absent, what will going to happen, etc. Then structural gene ZYA, promoter, say sequences, operator, inhibitor, that is I, repressor protein I. Okay, all those things are coming. Then what are the methods to introduce alien DNA into host cell? Alien DNA means our interested DNA into host cell. What are the different methodologies what we can follow? So here, that is heat shock treatment, micro injection, bioelastic gun. So these are the techniques. Then write any 3 tools used in recombinant DNA technology. Heat shock treatment irbodu, micro injection irbodu, bioelastic irbodu. Adu 3 namige methods agi. We can use these things as methods to introduce alien DNA, our interested DNA, into host cell. And the next question, write any 3 tools used in recombinant DNA technology. That is tools. That is restriction enzymes, vectors, host organism, then polymerase enzyme, ligase enzyme, bioreactor. These are the tools in the case of recombinant DNA technology. Okay. And the next is part section 2nd. Once again questions are there. So here you have to answer for 3 questions. 1, 2, 3, 4, 5 questions are there. Let us complete that one. Okay. So now let us have the next question, 33. So here I am taking only Karnataka PU board exam related points. Okay, it's not ICSI or CBSE. This is NCERT book based questions under Karnataka board exam. Okay, second PUC biology. So 33 questions. 33, yeah. In that first one, where the opioid receptors located in human body? Opioid receptors located in human body, in the central nervous system and gastrointestinal tract. Okay, opioid receptors. Opioid, after opium consumption, and this will going to affect on which part? That is the receptors present in the central nervous system, CNS, brain and gastrointestinal tract. Then mention the technique involved in cancer detection and diagnosis. Biopsy, histopathological study, radiography like X-ray, CT scan, that is computerized tomography, MRI, magnetic resonance imaging, then antibodies against cancer specific antigen. So these are the techniques with respect to identification of the cancer and detection, diagnosis. Then explain briefly the uses of transgenic animals. So transgenic animals, normal reasons for creation of transgenic animals. We can ask like that. Reasons for creation of transgenic animals or uses of transgenic animals. So biological product, study of disease, normal physiology and development process, vaccine safety testing, chemical safety testing. So for these things we are using transgenic animals. Then 35, mention the possible genotypes of blood group A and B. It's very simple. IA, I, IB, and IBI. So small letter I, if it is there, then it is called heterozygous A or heterozygous B. If it is IA, homozygous A, IB, homozygous B. Then explain briefly sex determination in honeybee. Direct question, isn't it? Yeah. And discuss the role of microbes as biofertilizer. So microorganism in human welfare chapter. In that rhizobium irbodu, cyanobacteria, azospirilla, mycorrhiza, etc., etc. They are utilized as a source to increase the fertility of the soil, isn't it? Nitrogen fixation, etc., etc. Assimilation of nutrient, etc. All are coming under that one. Then mutual relationship is also there under that, isn't it? Mycorrhiza, etc. Last question, that is 37th. What is commensalism? Under biotic interaction between the organism, commensalism, amensalism, mutualism, parasitism, predation, isn't it? In that commensalism is what? So here this is the interaction in which one species benefit, the other is neither benefited nor harmed. Then mention any 4 interactions of organisms that represents the commensalism. Orchid growing on epiphyte, growing as an epiphyte on the mango tree, mango branch. Clownfish living among tentacles of sea anemone. Barnacle on bark of the blue whale. And cattle ergot, that is a bird, and grazing cattle. These are the examples for our commensalism, isn't it? So by this, this is the last question, am I right? Yeah. So if you want more, you can use the plus subscription, where we are going to focus on Karnataka CET exam, and also we are with the detailed theory, MCQ, doubt clearance and mega quiz. This is the price value. So during the subscription, use my code KCET Murali and get 10% off regarding with the price value. Okay, this is a free practice test, how to do the test, etc. So thank you. See you again in the next class. Next class will be on 27th. Time is same under YouTube. Time is same, that is 9 p.m. and once again we are with the discussion of third set of questions under board exam, important questions under board exam, second PUC biology, set number 3, third set, and along with that I am focusing on important diagram based questions. So thank you.